Welcome dear students to a very important metabolic pathway, and that is the glycogenesis. The word glycogenesis has two portions. Glyco means glycogen and genesis means synthesis. So glycogenesis is the biosynthesis of the glycogen. You can define the glycogenesis as the formation or synthesis of the glycogen from glucose units. Glucose is a small repeating unit which is a monomer, and when these small repeating units are joined together in a large number, then you will get a polymer, and that is the glycogen. Glycogen is a homopolymer, means it is made up of only one kind of the monomer, and that is the glucose. Glycogenesis mainly takes place in the liver cells and in the muscles. And my dear students, the large amount of the glycogen is stored in the liver and muscle cells, but very small amount of the glycogen is stored in the kidney cells and in the brain cells. When a person is engaged in a very intensive muscular exercise and he needs a large amount of the energy, then my dear students, the glycogen which is stored in the muscles through glycogenolysis, it is converted into the glucose, and that glucose through cellular respiration produces chemical energy in the form of ATPs to provide that energy to the muscular cells. And the glycogen which is stored in the liver, this glycogen basically is broken down into the glucose units in case the blood glucose level of a person decreases. So when the blood glucose level decreases, then the glycogen stored in the liver is broken down to the glucose, and this glucose is supplied to the blood to maintain the normal sugar level in the blood for the normal homeostatic functions. And my dear students, similarly, the kidney cells also provide the glucose on a condition when a person is suffering from severe hypoglycemia. And in case when a person is having hypoglycemia or low blood glucose level, then the glycogen stored in the brain cells is converted to the glucose, and that glucose is used by the brain cells for their normal functioning, as the brain cells can make use of only glucose for the production of the energy required for their processes. So the glycogenesis takes place in the liver and muscles, and the large amount of glycogen is stored in liver and muscles, and small amount is stored in the kidney and brain. As far as the subcellular side is concerned, the glycogenesis takes place in the cytosol of the cells. It takes place at the rest condition after Cori cycle, and when the blood glucose level is very high, then insulin signals the liver to start the glycogenesis and consume the extra glucose molecules present in the blood. In this way, the high blood glucose concentration can be decreased by the process of glycogenesis. The purpose of glycogenesis is basically the storage purpose. My dear students, the glucose is converted into the glycogen, and glycogen is the stored form of the carbohydrates for the animals. And when the more energy is required for the different physiological processes, then the glycogen which is stored in the animal body, that is changed to the glucose, and then glucose provides the necessary energy actually. So the glycogenesis has storage purpose, and glycogen is the main stored form of the carbohydrate in the animal body. This was all about the introduction of the glycogenesis. Now let's see how this glucose conversion into the glycogen takes place. This one is the alpha D glucose. It has 6 carbons, and I have numbered them from 1 to 6. In the first step, one adenosine triphosphate is changed to one adenosine diphosphate, and the phosphate group released here that gets attached with the carbon number 6 of the alpha D glucose, and the glucose is changed to glucose 6 phosphate. This step is called as the phosphorylation of the glucose, and it is catalyzed by an enzyme hexokinase, and in the liver cell it is called as the glucokinase. Then the glucose 6 phosphate is changed to its position isomer, that is glucose 1 phosphate. In case of glucose 6 phosphate, the phosphate group is attached to the carbon 6, but in case of glucose 1 phosphate, the phosphate group moves to the carbon number 1 of the glucose. And my dear students, when glucose 6 phosphate is changed to glucose 1 phosphate, during that conversion an intermediate is formed, that is glucose 1,6 biphosphate. So you can say glucose 6 phosphate is changed to glucose 1 phosphate by passing through the intermediate, that is the glucose 1,6 biphosphate, and this reaction is catalyzed by the phosphoglucomutase enzyme. Now this glucose 1-phosphate, it is converted to a sugar nucleotide conjugated molecule, that is the glucose UDP, glucose uridine diphosphate. Look at this structure carefully. It is uridine triphosphate. This one is uridine, and these are 3 phosphate groups. My dear students, the 2 phosphate groups get detached from this uridine triphosphate. This breakage of the phosphate bond releases a large amount of the energy which is used during this reaction. And my dear students, now these two molecules, they will join together in this way. Like this. And now this complex, it has a sugar which is glucose, and this one is the nucleotide portion. So this is a sugar nucleotide complex, and that is called as the glucose uridine diphosphate. This one is glucose, this is uridine, and these are 2 phosphate groups. So it is glucose uridine diphosphate. This reaction is catalyzed by UDP, glucose pyrophosphorylase enzyme. And my dear students, now this glucose UDP will act as a carrier, and it will help in the joining of the glucose molecules to form the polymer. In the next step, now the uridine diphosphate means this portion, this complete portion. It gets detached, and now my dear students, an enzyme glycogenin, it provides a base and to join the glucose units together. My dear students, the glycogenin enzyme is actually the enzyme which will polymerize the first few glucose molecules, and it will form a small chain of the glucose molecules that will then trigger the next enzyme, that will be the glycogen synthase. So this glycogenin enzyme, it is a dimer. It has tyrosine amino acids in it, and this OH group is actually the OH group of the tyrosine amino acid. My dear students, the glycogenin joins the glucose units through this glucose UDP, and then a small chain of the 6 to 8 glucose units is generated, and in this small chain, the glucose units are attached by an ether linkage, and this ether linkage is called as the glycosidic linkage. Ether linkage is carbon oxygen carbon linkage, and in this case this one is the carbon, this is oxygen, and this is again carbon. So it is carbon oxygen carbon linkage, that will be an example of the ether linkage. And ether linkage, when it is specifically present between the monosaccharides, it is given a special name, and that is the glycosidic linkage. And this linkage is formed between carbon number 1 and carbon number 4 of the 2 glucose units. So that will be called as the 1,4 glycosidic linkage. And in this way, a small chain of the 6 to 8 glucose molecules is generated, and this small chain now triggers the next enzyme, that is glycogen synthase. And my dear students, this small chain will act as a primer. It will trigger the glycogen synthase, and this triggering will add further glucose subunits to this primer, and a long linear molecule will be created. This one is the linear glycogen, in which all glucose units are attached in a linear fashion, and my dear students, the linkage is 1,4 glycosidic linkage. Then the branching enzyme, which is specifically called as amino 1,4,1,6 transglucosylase, this enzyme now triggers the branching to this linear molecule, and my dear students, now the branching starts. And during that branching, when a branch of the glucose units is attached to the main stem, the glycosidic linkage is the 1,6 linkage. Means if this is the main stem and to this a branch of the glucose is attached, then my dear students, this attachment linkage here, that will be 1,6 linkage. Mean this ether linkage will be between carbon number 1 and carbon number 6 of the glucose molecules. But all the glucose molecules which are attached in a linear fashion, they have 1,4 glycosidic linkage, and this attachment here, that will be 1,6 attachment, and the remaining all molecules of the glucose, they will be having 1,4 glycosidic linkage. And my dear students, when this branching takes place, then the further process keeps on going, and the elongation of this molecule takes place with the help of glycogen synthase, and branching is further carried out with the help of this branching enzyme, and then finally you will get a very large elongated branched molecule of the glycogen that will be having a tree like structure, and it will be a macromolecule, that will be a polymer which is formed by the joining of the small repeating units, and those small repeating units are the glucose units. Once this glycogen is created, it is then stored in the liver cells to maintain the normal blood glucose level. It is stored in the muscle cells to provide the energy in case of intensive muscular exercise. It will be stored in the kidney cells again to give the glucose in the condition of hypoglycemia, and it will be stored in the brain cells to provide the energy when the person is suffering from the low blood glucose level. So, in this way, this glycogen is a stored carbohydrate in the human body and in the body of the most vertebrates. That was all about the glycogenesis. I hope so that this lecture will be very useful for you people. In case you find any problem, don't hesitate to ask. I will answer. Till then, take care.